Welcome to Examining Origins. I'm here with Joel, who is in the fields of bioinformatics and computers and science and technology, and he's going to talk to us today about the technology of DNA. So, welcome, Joel.
Hello, thank you for having me.
Now, I've heard that DNA described as like the most exquisite code of any code that we've ever encountered.
Yeah.
So, would you say that's true? And if so, why?
Yes, it is definitely the most compressed, most what we call eloquent code. It's um, we're reverse engineering DNA code to help improve our what I often describe as caveman by comparison code, the best leading edge technology I've ever seen or had the privilege of help. I've helped written AI for the Falcon 9. I've helped do all kinds of. I just finished a contract using AI to help better identify cancer research, all kinds of stuff. All that is caveman. That's just literally smacking sticks against rocks compared to the beauty of DNA code. It's what's called a universal language code. I can go to the depths of that, but it's also embedded on a literal molecular atomic scale. So I started my civilian side of my career in nanotechnology, trying to develop MEMS and micro machine type stuff, for semiconductors and microchips. And so I've, enjoyed trying to make our, again, caveman human technology, small and precise. And you try to compress it, using all different types of protocols and codecs and stuff to make it as efficient as possible. DNA blows it all away. We have to develop groundbreaking, revolutionary new technologies in our code base just to begin to understand seemingly basic things in DNA, if that makes sense. DNA teaches us.
Can you give me some examples of that?
One might be, everybody runs an antivirus program on their computer, for example. Most don't really know how it works. They're just glad that it does. It doesn't. It's not always foolproof, but it's pretty amazing. A lot of the high tech stuff that I do, you can't do our work without some of the most expensive embedded antivirus detection and network protocol security stuff. You just can't do our work without it, because one wrong slip, of illegal or whatever unauthorized code, and just not only do you lose secrets, but it destroys your work. So DNA has automatic antiviral code built into it. Literally, it has line one retrotransposons that there's these enzymes that used to be called jumping genes, right? When it was first discovered, guys like me, we thought, oh wow, DNA, like parts of itself breaks off, floats around randomly, lands on other pieces of itself, and breaks things, right? It just starts editing things and destroying the code. How the heck does this thing ever survive? Further research discovered it's not random. So the code is designed to separate these enzymes, travel along the source code chains, find where it has some way of that's the hard part. How does it know to detect where the errors are to begin with, self corrects itself, and then has these pieces of retrotransposons designed to separate from the code, float around, not aimlessly, specifically land on where the errors are and fix it. And then again, that's just the autocorrection. The antiviral code. There's R-type processes where these transposons and retrotransposons they actually are designed to move inside and outside of the nucleosome, right, through the pores of the nucleus, and that's designed to transfer code that way, particularly mRNA and other critical information telecommunication processes. And so DNA has embedded functions to scan basically what these files, it's literally like a file, these strands of RNA and other things, to see if they're correct, see if they're authorized or not. And if it's a virus, the viruses that there are retroviruses and things that are natural, they're seemingly natural. That also the humans, man-made retroviruses, they have to be very cleverly designed to know how to sneak past these antiviral DNA programs, these antivirus programs. trying to see if I can help it make sense in normal computer terms.
Yeah, I mean, it's amazing. Now, so it's interesting because you keep using the word designed.
Hmm.
And
Yeah.
so how do we know that, like, so a lot of people have a problem with using the word designed, but it seems like these are the types of systems that we know as designed. We don't have material processes that can produce these kinds of things. So, what would you say to someone who who is like, well, I'm hearing what you say, but then I I hear all of these scientists, so many scientists that say this all happened by material processes. Amino acids came together, formed proteins. All of this happened with time and chance and the chemistry.
Once upon a time ago, many billions of years ago. Yeah. Basically because okay, so there's only really two dichotomies, I guess. Design is where you've got a system that works for a specific purpose, for specific reasons, through specific actions, like code does. Non-design would be, pseudo-random, ad hoc stuff that just happens by accident, right? And there's nothing in DNA, nothing in biology at all that ever happens by accident. Not even mutations. Mutations don't even happen by accident. There's specific reasons why certain mutations seem to take hold and why others don't take hold. There are, like I said earlier, error correction code within DNA that detects and finds and removes mutations, aka code bugs. So, and all these other types of things I mentioned, the retrotransposons, the input outputs of the computer kernel itself are all very highly specific. They are designed to allow and disallow certain things. So that is by purpose. That causes, and in fact, the entire DNA chain is literal instruction code that is designed to activate all of these systems, all of these functions. That's by design. It's never by accident.
You mentioned transposons or retrotransposon. Could you explain more about that?
Basically, it's code sequences that are again designed to transverse or move around, so to say, the rest of the code, to put it as simple as possible. Examples are mRNA is a transposon type information that leaves. Some of it does come back into the nucleosome, but most of it is to go talk to the rest of the cell floor subcomponents like the ribosomes and other things that the original source code, the DNA source code, talks to. There's telecommunication going on. It's a literal computer network, and then there are just bits and pieces of other information. Uh, there's classified as R-type enzymes in the nucleosomes and the nucleotides that move back into the nucleosome. The COVID-19 vaccines, for example, they're classified as mRNA vaccines. Those were designed to try to, they were mostly trying to design just to hijack the ribosomes to create uh, antivirus specifics to the COVID virus. But there was indication that some of those did form into retroviruses that did bypass the R-type protection processes from the bits and pieces of the artificial man-made code that was trying to make the artificial, I like to call it, a genetically modified vaccine. So those pieces would be another example of transposons that sneak into the DNA, and we're still hoping, quote unquote, that it doesn't cause lasting harm.
Oh, wow. Yeah, that's really interesting. Okay, now there was something else I wanted to mention because you mentioned mutations. Even mutations
Mhm.
have a purpose, and that's something interesting that I've been seeing in some of the research is that actually we see that the the cells themselves like make mutations happen, intentionally make mutations happen when they want to bring forth something in the genome. And so, can you tell us more what you know about that?
I can. In fact, I'm thinking how even the way I described it is kind of a misnomer. So when code modifies itself, it's not by accident. It's extremely complicated. And so examples of that are with, gosh, there's so many examples my brain is going through. One of them is in how DNA is a literal database information archive retrieval system, right? You've heard of inactive versus active genes. Well, inactive genes become temporarily active. They literally, the code unfolds itself. It's by program. There's enzymes that travel along. Hey, like the liver cell, for example, all cells in a eukaryote like a human have identical code, right? So your brain cell uses the same exact code as your heart cell or your thumbnail cell. But certain parts, certain functions of the design, like your liver cell, for example, is a great example. When you ingest something that's unhealthy, like a toxin or something, you've got other systems in your system that identify. Say, hey, look, you just ate, some old ketchup or something. This is going to hurt you, right? And so it activates all these other systems, all these other telecommunication networks to tell your liver cell to, hey, we got an incoming toxic coming down, the gastro whatever intestinal tract. Get ready, right, to attack this toxin. And so the liver cell goes and scans its own code, which is identical code to everywhere else in the entire system. Says, okay, we're about to have this specific toxin. How it communicates this to all the different cells is another mystery. But it specifically says, I need to get the code for this toxin. I know what this code is. It's here. It's in this address location within the database, right? And so all these chemicals react on the DOM and DNA, which is a three-dimensional structure. It's a physical structure, right? All these 4 bits of chemicals all form in these tightly, it's a whole other topic. And so it unfolds parts of the inactive genes to expose it so that the retrieval systems, the code readers, so to say, will go across it at a specific time in a specific place in a specific way, in a designed way, to read that archived code that's stored in the database to build the necessary proteins and other structures to fight that toxin. And many just untold other things that the body does every day. And then when it's done, when it's built enough of these toxins and the body says, the rest of the body signal, the internet inside your body says, okay, this toxin's done, you're okay. It refolds or re-archives that data, that information, that part of the code back into the genome, back into the, it's amazing. It's so freaking amazing. It would be beyond, I think, a miracle, I think, for us to develop that for our own code, which is what a lot of us are trying to really work towards. But I forgot your original question.
I was talking about mutations
Yes.
and how we were finding that they are intentional, and asking you to give me like what your understanding of that is. So you mentioned the unfolding, the finding of the right program or the right, the section of DNA that's needed.
Mm-hmm.
But what about, for the liver example, but what about with the liver example, is there something that's changing the code for a particular toxin, or is that just a separate thing? What I mean is, was that an example of an intentional mutation, or just an example of something awesome DNA does?
Right, and that's why I describe it as kind of a misnomer. So when something's intentional, then it would be probably wrong to call it a mutation. If you want to define a mutation as something that happens by accident that was not designed, which is the premise for the evolutionary theology, was that something that was not intended makes a change. And so that does happen. There are random mutations. And there's all kinds of examples of how a lot of the presumptions of known, scientifically observable mutations, like the citrate mutation, for example, in E. coli, we can go to that, how that was falsified, which is another fascinating topic. But DNA has known weak spots, basically. So there's intentional changes, basically. The brain cell, for example, there's some people call a fourth dimension to the three-dimensional code array of DNA, where the DNA is designed to change itself over time. So time being that fourth dimension. So there are programmed code iterations where the code is designed to modify itself as you age. and there's all kinds of awesome work going on with that. But there again, what I would describe as weak spots within the DNA code itself, because all code is subject to bugs, right? No matter how beautiful your code detection and antiviral, whatever protection algorithms you try to design to try to preserve your code, mistakes are going to happen, right? Code is extremely complex, especially complex code. More, in fact, that's the basis of chaos theory. The more complex a system comes, the faster it tends to break, right? So DNA is extraordinarily complex, and so the DNA though is designed so it's got known soft spots, weak spots, whatever you call it, where uh, common breaks will almost always happen or will routinely happen, and the DNA tries its best to fix those mutations. There's backup routines for if it can't fix a random mutation, it'll try to augment it, try to do something else to still either create the similar function or a different function. That it's just so complex to try to continue to do the overall system design of staying alive, of being healthy. And so even accidents that do sometimes don't get fixed. In fact, that's the way I like to describe it as, biblical science describes how, mankind broke the creation by, violating the OEM recommendations, I like to put it. So the code is breaking. Our DNA and everything around us is slowly breaking. And so even the auto correction features designed into DNA are not 100% anymore. And so that's where death and disease comes from, these code breaks. And so another example, longevity. If DNA did not break, there's all kinds of different theories with that, then we, in theory, live forever. The code would continue to go on, continue to replicate itself, and you'd never die. So there's designed obsolescence. Just like in the SpaceX Starlink system, there's designed obsolescence so that you don't have to continue to figure out how to pull out the obsolete stuff when you want to replace it, upgrade stuff. DNA does the same thing. There's weak points where DNA allows itself to be damaged, so to say, or prepares itself to be damaged. Sorry, to replace or augment or redesign, even sometimes. And that's the other fascinating thing about DNA I haven't mentioned yet. There's literal machine learning models, we call it, in DNA. And so it's designed to basically test for these errors, test for these mistakes, as well as designed variants to then make corrections, to make new models based on the environment given. That's what we call it. Biology calls it adaptation. It's exactly the same thing in computers.
Yeah, that's what I've been learning, that a lot of organisms are able to adapt to their environment, not over millions of years,
Right.
but even over their lifetime. That
Yep.
basically they're within the cells, they can recognize that there's something in the environment that's needed for that particular organism, and then it like pulls out that code that maybe was hidden in there, like you mentioned earlier. So there's been some morphological changes observed in fish and lizards, whereas like they were put on a different diet, they evolved. I mean, well, I say evolved because that's what it's promoted as in the
Right.
literature, but basically they, in the lifetime of a single creature, formed a cecal valve, and then when they were placed back on a different diet, removed the cecal valve. So a major change in morphology of the creature, went forward and backward in the lifetime of the creature based on the environment it was placed in and what it had to eat.
Right. No, that's exactly what falsified the LTE experiment, if you're familiar with that.
Or is this the Linsky experiment, the E. coli? Yeah, I'm glad you came back to that, because I wanted to ask you about the falsification. Because my understanding is that basically these creatures like the E. coli, they like changed to a mutation, caused them to be able to metabolize citrate
Mhm.
where they couldn't metabolize citrate before. But then, like you said, maybe mutation isn't the best word, because it seems like an intentional change in like that was already possible within the genome, like a change was made, and this is repeatable, like it's been repeated over and over again, that if you put those E. coli in that environment, it will always like pull out the ability to metabolize citrate. Am I understanding that correctly?
Very close. Basically, what you're describing, what this article describes, this is one of the many falsifications, was that the mutant that they originally thought from the Linsky lab was not a mutant. It was not a mutation. It was what's called a promoter gene. Somewhere in here is a promoter. Hold on. Here. So the code change that happened that allowed it to process the citrates that you accurately did describe was designed in the code. It was a design feature. So when the organism detected the citrates in the environment, which is not optimal, it prefer not to have to consume citrates, but when it has to, it's designed to switch a switch, basically, to make a trigger, promoter capture, they call it, where the code, it's like an if-then statement, basically, except far more complicated. The code changes itself to start the process to eat those citrates, basically, to breathe the citrates. And this experiment found out that instead of the 30,000 generations that Linsky thought it happened in, it happened within 12. It happened within, and there's been further papers I've read. They found it, like you said, happened within the same lifetime of one cell of an individual bug would do it. And then when they took out the citrates, it reverts back. The if-then sequence trigger undoes the code change, and it doesn't need to process it because it's inefficient. The citrate processing takes extra energy. It's harder to do. So the software design, the system, the computer would rather not waste program cycles trying to process citrate. So it undoes that change. And so like this, many of the other articles talk about how there's no new genetic information, no novel gene function evolved. That was never a mutation and nothing evolved. It was designed that way.
Okay. Well, so basically, when you say falsification, you're saying it's falsification of the idea that mutation plus natural selection produces new genomic information.
Right. So the evolutionary theory would claim that something random on your computer, your maybe Microsoft Word you're typing, and all of a sudden when you type the letter J, some funky character comes up that doesn't exist in the alphabet, right? And every time you hit J, it always does that. Oh, that's a natural cause random mutation, right? But that's not what happens. It's not in this experiment, not in anything else, these other experiments. What's happening is if if you change a setting on your computer and you want your J to look like an I, for example, if you're gonna try to type something in Latin or some Chinese uniform character or whatever, that's programmed by design to change that input, that key that you press, to something else instead. That's what's happening in biology.
So can you explain the concept of a promoter?
Yeah, my simplest way I've tried to help explain it to fellow IT guys is so like Ethernet. Most people, maybe, I don't know, a lot of people understand how Ethernet protocols work, TCP/IP, PPP type internet protocols. And I guess I could try to give a quick rundown of it. So everything that we do on the internet, this video, for example, our voice, everything, all the pictures that we send, all our cute cat pictures and everything, the pictures themselves aren't sent, of course. Everything, and stop me if you know what I'm talking about. I don't want to take you or your audience's time, but your cat picture, your kitty picture, is broken down into packets, right? So it might be a megabit of data that makes your picture. The computer will break it down into thousands or however many sub packets of the information, and that's what gets sent across the interwebs. All the other computers that translate all this stuff and move it all around, and eventually goes to the source of who's watching, who wants to look at your Facebook post. And then your computer takes all these packets from all over the world sometimes and puts it back together. And here's your picture. Well, on each of those individual thousands of packets for each individual simple thing like that, much less the complex information we transfer like this, has what's called header and footer packets, or tags. So each packet, each individual packet, like you can also describe it as a car driving down the information superhighway. Some people try to simplify it as there's a front part, a header, it's called in IT terms, that directs all the other computers it touches. Okay, this packet, this is packet number 162 out of 2012, whatever, is going to this region. So the gateway that sees this packet says, okay, well that direction that you want, that this packet wants to go in, is probably down this area, this road, down my spanning table, my routing table, whatever you want to call it. And so I'll send it that way, and then the next computer gets it. Oh, you want to go there? Okay, you probably can get there. And it does this. Who knows how many times? They call it a trace route. Some can take a few dozen. Some can take hundreds, depending on all kinds of different things. And then when the packet with the header reaches its destination, oh, you want to come here? Okay, then that computer takes the header off, and then it combines the individual packets. Like I said, makes your kitty picture. They also have what's called footers on the end of every packet that also has information specific to where it came from, and basically what all kinds of different information for all the computers that ever touch it, to know exactly where, what, when, how that packet is supposed to be recombined later to transmit what we're doing. Again, sound, video, everything. And so DNA or biology does the same exact thing. When different chemicals go across the DNA source code, it's by design, it finds the pieces that are supposed to be transmitted, and it has a specific mechanism that will lift the code off the DNA. I forget the term for it in biology now. I'm trying to remember. There's a specific term that makes the RNA copy, or the DNA copy, that turns into mRNA. It is programmed to know which pore to go out when of the nucleosome, and all kinds of other things get attached to it. It's also included it's activated by what's called a promoter gene region. And they also have, which is a header. They also have footers, which are called terminator gene regions. So that all the different the machines, all the different manufacturing machines in the rest of the facility know when, where, and how, and why, which piece of DNA packet is being used in what for. Like with a ribosome. Because, oh, you want to go to a ribosome. Okay, so we're going to wait till you connect with the right ribosome at the right time. Okay, we're going to wait now for the tRNA to literally decipher, because it can't do anything without the key, the cipher key, which is another computer function, to be able to go through the ribosome to mix with the amino acids and create the actual protein strand, which there's all kinds of coding for the protein to fold, which is a whole nother miracle, a whole nother huge topic by itself. But that's what a promoter gene basically is. Promoter region. It's designed to help the computer system of biology know where something starts and where it's supposed to go. Literally, just like Ethernet.
Wow, that's amazing. Now, did we know that about DNA when we started doing this with computers?
I don't think so. I don't know. I don't
Okay,
really
I
don't think.
was just
I
wondering
learned in
if
the
we
90s.
came up with the idea because we learned about it from DNA, or if it was just like a surprise that like there's the same function there that is,
Yeah.
created by humans as well.
Maybe I don't know everything obviously yet, but I learned about Ethernet packets and TCP/IP stuff in the 90s. I've written my own internet protocols and stuff over the years. I didn't know what promoter genes and stuff were probably until about 2008, 2010-ish or so. But that's because I hadn't started really into bioinformation stuff yet. I looked at the way biology does it, and hey, that's a header packet. So it just clicks, And since then, everybody I've worked with now, there's academics that will disagree, of course. That, oh, it's not a header packet. This just it's something else telling the computer, or telling the non-computer where code starts and how to transmit it. I'm like, okay, well, I mean, it's the identical definition and function of an Ethernet header packet that a promoter region does. It's exactly
Well,
identical.
now, since you brought out the people that you work with and the people in, academia and stuff, so you are working around a lot of people with scientific knowledge. And I often hear, that like oh, so many the vast majority of scientists fully embrace evolution, molecule to man evolution. So I want to ask, is that your experience?
Hmm. I don't know any scientists that believe in evolution anymore. That's a religious tactic. I like to remind people that's what Galileo Galilei faced when he was trying to convince the Vatican that Copernicus had a point, that the planets revolve around the sun. Well, no, the peer review scientific community of his day said no, it doesn't, because we said so. It's appeal to authority. It's appeal to popularity. Science does the exact opposite. Science, when Bacon invented the formal scientific method, it was specifically to avoid it, and prevent what the Vatican did, what those people you're talking about do today. Because he even Bacon, for Sir Francis Bacon, if you ever studied his story, it's amazing too. He wasn't really a scientist; he was more of a politician, right? He was a statesman, in the Anglican Church, and the Anglican Church at the time started making all these policies, trying to say, well, the Bible says this, and Bacon and others like, no, the Bible actually doesn't say that. That's a whole other topic. But so Bacon developed this method. Say, like, let's use empirical evidence. Let's actually look what the Bible says, with examples, with observations and counter observations, whatever we can, to confirm this is what the Bible actually says, and this is what God actually says, basically, and test it. Create a method to test these things, so that the scientific community or whatever might be wrong gets corrected with. And so those that say that, oh, the scientific community says something, hate science, in my professional scientific opinion. It's
Okay.
purposeful bias. I love how Einstein puts a lot of things, but he explained how science and religion are required together. They're interdependent to come to search for truth. I love how he puts it that way. It's not science or religion or whatever, or even academia. It's just bias. People often want to be biased. They think that they're right. They develop an entire ideology around something, and because of cognitive dissonance or whatever other human frailty, we just can't be wrong. So that's what science was developed to test, what you think might be true. And that's the other thing. Those of us that I work with, we always recognize science can't prove anything. So you need philosophy or religion or whatever you want to call it to test your scientific observations. That's the logic of things, is what helps try to develop proof. And you add the scientific observation. Science is just a way to observe things and categorize things, make repeatable observations in real time, I like to say. So those that think that science does anything else, they're just not using science. And that's the problem. So in my work, in my career, all of, I don't know, the I don't know how to put this easily without sound like I don't want to brag, but the best teams I ever worked with, we always challenged each other. You could say 8 + 8 is 16. We would challenge that. No matter how dogmatic. So we used to tease. You know, we used to go to college classes. Work for SpaceX, and we would try to teach this philosophy to students. Say, hey, even if you know for a fact, quote unquote fact, that the force of gravity is 9.8 seconds per meter squared on the surface of the Earth, right? And you've tested it 5 billion times over 2,000 years. You know for a fact what gravity should be, right? Always test for it. Before you do your rocket launch, test the force of gravity at ground level or sea level before you launch, just to make sure something didn't change. That's science.
Okay, interesting. Now, let me ask you, I want to go back to the protein folding, because you said that's a whole nother
Oh my God.
story
Yeah.
about the parts of the DNA that direct protein folding, or I don't know. Why didn't you describe what you meant?
So protein folding is a whole nother ball game. Are you familiar with AlphaFold?
No.
AlphaFold 2.0. It's a organization. It's a nonprofit. Amazing group of researchers. They have used very advanced AI to predict and actually basically watch how protein folds are made. Right. So in the past, when the protein chains are formed outside of the ribosomes in biology, it's just magic. It's okay. It just does it. It's programmed that way. It's designed that way. These atoms within the proteins are so carefully, precisely arranged. Either by accident, some people want to presume, or whatever, to start to once it leaves the ribosome, it doesn't just come out straight. It comes out and it starts to fold itself in a very specific programmatically way to be able to do the things that these protein structures do, from everything from creating new ribosomes to creating new nucleosomes and membranes, all the lipids, all of the thousands, if not hundreds of thousands of processes within a single cell, all come from these proteins that fold themselves, right? And so each type of protein can be folded into all kinds of different configurations. And it's infinite. It seems it's just unlimited amount of configuration for these basic building blocks of life. And up until recently, it was just beyond human fathomability. We were just okay, yeah, it obviously does it. We can see how it does it, but we don't know how or why it does it. So now companies like AlphaFold, they have been using computers to try to predict how these proteins, the molecular force and the barriers and all the different chemical reactions within these atomic structures fold. So you have to put the exact chemical, the exact atom, the exact ion with the exact position. You have to do everything. It's like playing with a very complicated Tinker Toy set. If you have to put the right thing at the right place at the right time for the next thing to connect just the right way, and then for it to all to move together. It's just mind-boggling. And so last I read a few years ago, they came up with like 400 million protein folds that they were categorizing, and they were making it public, for other researchers to try to help determine, because that's the only way you can really try to understand DNA code is you watch how and where and try to understand why different protein structures form, to try to reverse engineer where that code comes from, where the code looks like, and then try to find, and then you scan the whole code through titration and other methods to try to find where the code might be to make that protein change. And it's extremely difficult, but we're doing it.
Yeah. Now, would you say this is a good analogy to this? Because what I'm thinking of as you're talking about these protein folds is like a car assembling itself. Like as if like all these car parts are coming out of the ribosome. Of course, this is an analogy, but like the car parts like coming out of the factory and then like putting themselves in the shape of the car.
Yeah, I'm trying to think of another real world example. I've messed with so many things. What else have I messed with that self-folded itself? I mean, there's certain plastics and polymers that do it by design, but those are so simple. I'm trying to think of a good analogy.
Well, yeah,
Nope.
I mean, and of course, a car doesn't assemble itself. So
Right?
I'm trying to say, like, a life does something amazing that we don't have a corresponding activity, because I think
Not yet.
don't the plastics, it's done, I mean, it's happening by a chemical process naturally, but this is something that's wouldn't happen unless there was a specific design involved.
Yeah, in fact, you're reminding me. The example I like to give in a lot of conversations about this is I've got a wonderful picture from my old job back at SpaceX, where Lisa was standing between. Let me see if I can share this. Between what we call our Merlin M1 rocket design. That's basically an atmospheric configured Merlin engine that you use for this first stage when you lift off, and next to what's called an MVAC, Merlin vacuum engine design configuration, that's used for the second stage out in space. I'm trying to share it. So on the left is the atmospheric. This is what we use on the first stage to help lift off from the ground, right? And it's more compact. It looks simpler, but it's not. They're actually basically the same. But the nozzle and other engine components are designed to be more efficient at sea level, so you can lift off, and then once you're in orbit, the first stage separates, and SpaceX is remarkable. We actually recover our first stage components, and then the second stage Merlin, again, if you can see the similarities in the piping, they both have gas generators and all kinds of similar piping, but the nozzle and some other components are more differently designed to be more efficient in vacuum or near vacuum in orbit. So because this engine, this will work in orbit in the vacuum of space, but not as efficiently as this design, right? So our hope as rocket scientists someday, and Elon's trying to work on it now with the new Raptor engines, is that we will develop the correct materials that can withstand the pressures and the heats, that can automatically transform itself, reshape itself, refold, whatever you want to, how you're going to describe it, to switch between the different configurations. It's not impossible. It's just a matter of discovering the right materials and the right software, the right hardware to do it. That's what happens in biology. You take one configuration and you can programmably make it fold into something different using the same materials. These are all the same materials. Same basic design, just slightly different. I mean, it looks different, but in theory or in practicality, it's just a different efficiency for whether you're in an atmosphere or not. Same thing in biology. There's different things that are different efficiencies, like the citrate example for E. coli. Yeah, I'm still sharing the universal information code graph for the kernel. We call it so every computer language has a code kernel, which is the core structure of how the information is translated and used within an operating system, or decompiled from the machine language, it's called. So DNA has that, where there is upwards of 40,000 possible amino acids that the currently known life forms could create using the processes in biology, and the known processes in biology. But there's only 64 of those up to 40,000 plus combinations used in all known life everywhere. And as this picture, demonstrates, microbes, all plants, all insects, all mammals, all animals, even human beings, everything in life, from the smallest to the biggest, uses this kernel.
Now, a lot of people, I've heard them say that, like, this is evidence that we all came from a common ancestor, because, we're all using the exact same code. So doesn't that point to a common ancestor?
Even the common ancestor uses the exact same code everything uses still today. So there's no change in this kernel. The first thing I would say. The second thing, it's a common misnomer. The common ancestor mythology, that originally came from Christian theology. I don't know if you've heard of natural theology theories from like Lamarck and Bill Paley.
Yeah.
Yeah. So that idea, that's a Christian theology, theory that God created diversity in life from the common ancestry out of the Garden of Eden. That non-Christians, since took very, I think, I don't know, couldn't get into too many details, but very badly motivated motives to replace the Christian God for the same exact theory for what I call the personal God of natural selection doing it instead. Literally, some divine entity, they want to think nature did it on its own. Christians want to say God did it. Same exact theory, but even if they were, and they're not common ancestors, there's so many diversity, there's so much differences between what we think was some of the earliest life forms to today, that there's nothing common with them anymore other than the code language. The only thing common between everything is this kernel. That's it. It's
And
one
so,
computer code. Huh? Sorry?
do you think that just that it's based on these few things makes it so that it seems like this is a more simple process that could arise by natural, like by material, physical processes?
Yeah, no, not at all. Because not only did this simple version seemingly, this is not simple. This is extraordinarily complex, super precise, super compact. This is universal information. We call it in information science. I like to describe this as the holy grail of what we try to do in our industry, my profession. If we can develop something even remotely similar to how one code kernel can basically translate or emulate everything else, then that would revolutionize everything that we use for computers today. You wouldn't need Apple or IBM. You wouldn't need Azure or Amazon. You wouldn't need any of these other platforms. We'd have a universal code kernel emulation that everything could translate through natively. And that's what biology does. So not only is this basically impossible to do right now for human, motivations, but if again this was truly non-designed and was just going to happen, randomly or naturally or whatever people want to presume, then there would be a near infinite combination of mess. The other 40,000 amino types would be all over the place. Life would have all kinds of other, you would like to presume, even functional systems to translate from outside of this kernel, but we don't. And so, in theory, a biologist or bioinformatician, in fact, we do it. I have done it. We try to augment and create our own human-designed binary machine learning language to be able to literally upload into this quaternary machine learning language to try to better learn it, understand it, reverse engineer it, and ultimately to try to better adapt our code into this super advanced code, basically literally reverse engineer it. And so I'll make changes. I'll take a DNA strand of something that, either myself or others have tested the different protein functions and other things to try to identify what this code is supposed to do. And I'll change it. I'll upload, there's different CRISPR functions and other things you can use to upload your titrated binary based, translating into the quaternary bit based code, insert it into this pre-existing universal code and watch what happens. I'll make predictions. Okay, if I change this code to try to do this, hopefully this will happen. And it's an extremely delicate and time-consuming process, but we can test for it. And so the current processes that this software, this computer technology, this computer nanotechnology operates with doesn't change. It should. You would like to think, it starts to whenever mistakes happen, but it doesn't make viable changes. The machine learning that's in biology is a very constrained, very contained set of what we call finite state machine groups. Biologists call it allele groups, but it's just another computer code designed to allow for certain variation within the design to allow certain things like whether your eyes are green or blue, or whether they're, you've got certain muscles and certain groups and everything. That's all part of this finite state machine set. And if it goes outside of that, you won't have like natural purple eyes, for example, because it's not in the finite. It's not in the linked list, we call it. And so, you could try to artificially augment it. Some people have tried. They're trying to do that, trying to create custom eye color, for example, custom earlobe shapes. It's all kinds of experiments. Custom skin. People that want to pretend that they're lizards, for example, instead of getting all the tattoos and all the stuff they do to their skin, they want to see if they can create genetic, gene therapy to grow scales on their skin. In theory, it's possible. We have to learn where the specific, finite state machine group is that causes skin to be skin, and change it to something that causes a different computer's skin to be a scale. If that makes sense.
Now, how close are we to understanding that stuff? Like, how much of our DNA is still completely unexplored or not understood?
I think that near, if not 100% of the human genome is deciphered. Most of other organisms are, where there's wonderful gene banks out there that anybody can go into. So many are public, some are behind a paywall. But my experience on the bleeding edges of fields of technology is that the things that people that aren't in the field have been achieved 10 or 20 years prior. So I can give a few case examples, but I think this is already being done on an experimental level, and it's just a matter of time. There's what's called the technological S curve. When you first start a new idea, it slowly becomes popular. It's not really well known, and then you usually run into mistakes. Like the development curve goes backwards. You lose a lot of money. You have second thoughts and triple thoughts, and whether you're doing the right thing. And then eventually it starts to work, and then it becomes more well known, and then it skyrockets, right? It's like an S on a curve. And I would not be surprised if we're on the upper edges of the technological S curve in a lot of these artificial biological changes. So it's just a matter of time, really. It's well understood enough because it is a computer. Biology is computer technology, literally. Well, nanotechnology, but I've been working on human nanotechnology since 1999. 25 years ago. You know, we're learning. We're reverse engineering God's technology, whatever you want to call biology. And I've had a couple of hires trying to get me in to help develop. You've probably heard of biological like hardware storage devices. Trying to store human DNA or human information on DNA type devices.
To like preserve yourself for future generations, like preserve your, is that what it's for?
No, to preserve anything, any
Yeah.
data. Whatever, your cat pictures. Convert that from binary to quaternary, store it on this DNA optimized hard drive. There's ongoing technological developments for that today. This is years ago. NIH tried to hire me to write some sort of DNA archive software tool they wanted. But I didn't want to move to Maryland. And so there's all kinds of modern human-based technologies based on biotechnology. There's projects I could share about how researchers are editing entire organs genetically, through the universal language. There's projects where they're trying to develop like pig organs, right? Because a lot of people say, oh, humans evolved from apes. Well, maybe not necessarily. There's more genetic similarities between humans and pigs than there are other primates. So researchers are trying to basically augment pig organs like lungs and hearts and things for humans to not reject it, right? Even if you get another human organ transplant, sometimes the organ's not perfect. So your designed self-defense mechanisms and your computer tries to fight this foreign object. You have to take drugs and stuff to suppress your software protection programs, right? So the theory is if we can program a pig's lung or some other animal's lung to trick your self-defense systems not to reject it, and to also process human blood instead of pig's blood, because there are differences, then people that have lung disease or kidney disease, and there's all kinds of research out there to replace basically every organ in the body. Maybe not the brain, but I bet you there's somebody working on that too. To edit this like you do with any other technology.
There's a lot of like Christians who do believe in common ancestry, that we all evolved from a common ancestor. And so they would hear what you're saying and they would say, yeah, see what you're saying. We're saying, look, it's not by accident, but God instilled in biological organisms the ability to change and morph over time. And so we believe it started all with one form of life and then slowly developed. So do you think that's true, or do you think there's a limit to the ability to change?
Yeah. So again, the whole original theory of common ancestry is a Christian theological, framework. And non-Christians just stole it literally. But for me, as a developer and a scientist, engineer, and all that stuff, I would have to see examples of it, and I just don't. I mean, I've done a lot of research. I've tried to do it myself artificially, with very marginal, levels of success. But there's no example that I know of intermediary, whatever you call it, species. There's no example of fish turning into anything else not a fish. Even the mud. What do you call it?
Yeah, so what you're saying is like you have no problem theologically with the concept of God creating life with the ability to evolve and change, but it's not what you scientifically observe. You don't believe in that based on what you observe happening with living systems.
Right, because today, you know, the observations today prove otherwise. Because again, all of these different variations, these they're literally finite state machine groups that biologists call alleles. They are finite. They are very carefully designed to allow for a certain level of variation and no more. If mutations or whatever cause these FSM allele groups to go outside of those weighted linked lists, we call it in software, the program dies. You know, in biology terms, it gets diseased or you die. You know, genetic disorders, and so those limitations within the current designed literal machine learning adaptivity functions have you can we can observe the program limitations within those organisms. There's certain things that can adapt to live in extreme environments, you know, whether oxygen or temperatures, and certain things that can't. Other software designs don't have those same conditions. They have restraints to only live in a very specific environment. And so, yeah, I mean, if God can, and God could do anything. God could redo the entire thing of biology in a heartbeat. That's a theological question of the limitations of God. But as far as what we can see in God's designs or this biological technology, it is programmed to not be allowed to go beyond its machine learning range. If that makes sense.
Yes, and this has been very enlightening. I've loved this conversation. Thank you
Yeah,
so much.
yeah.
Is there anything final that you wanted to share with people?
Well, I mean, I got all kinds of other interesting analogies I can try to help picture the complexities and the what I like to describe as the impossibilities of evolution designing this stuff. Um,
Well, go
one,
for it. Anything
yeah,
you want to share. Yeah, go for it.
one such analogy I like to make. Well, it's not even an analogy; it's a literal comparison. So, like it's described earlier, how mRNA is a literal, you know, packetized codec information transfer protocol technology. It's literally like Ethernet, right? Complete with header footer stuff we touched on. So one of the popular ideas out there, what's called RNA world, you've probably heard of. Which, by the way, there's the scientific paper that started that like 12 years ago has been retracted. It has been falsified. And I've got the copy. I can show it. I can present that for those interested.
Wow.
Yeah, because nobody else was able to repeat the experiment that these alleged um, organic polymers could self-replicate. The theory that they do, because it does it in biology today, but they can't do it on their own. I got the papers to show the reasons why. So yeah, that was retracted like I don't know, 12 years ago. They retracted. People still preach about it. But anyway, so for RNA, for a packetized information protocol like RNA, to have evolved itself by itself, first of all, is stupid. Sorry, impossible. But the claim is that the Ethernet basically could evolve to create its own source code, which is crazy, to create something vastly more complex than the entire World Wide Web. And I know how the World Wide Web works, right? I know how gateways and routers and the difference between level one, level two routers and switches and all that stuff. I know all that junk, but it would blow my mind to know how complex the internet has become today, right? It's crazy. But something even more complex, more vastly complex than that, than the entire web, could evolve itself pseudo randomly by accident without computers, literally, no computers, and just here it is, transmitting all our kitty pictures. That's RNA world. It's that crazy. It's like no, I'm sorry.
Okay, so you're saying even the RNA is more complicated than the entire internet.
RNA, right? Oh yes, and RNA can't exist without DNA. The theory that RNA could create DNA, sorry, I'm sorry. That's like again, it's like Ethernet creating itself to create computers that could create internet transfer protocols. Just which is what RNA is. RNA is an internet transport protocol. It's just
Okay,
literally
why don't you explain a little bit more about that? Tell people what exactly is RNA. Why couldn't it come about by material processes on
Well,
its
first of all, because it's very complex information designed for very specific purposes. DNA does not just randomly spew out RNA all over the place, like evolution theory would suggest. It's very specific in it's a codec. It's compressed, decompressed information designed to activate certain hardware protocols all throughout the cell, all throughout the nanotechnology vectory. And so for that to have just randomly developed itself as a miracle on top of miracles itself, but there's no examples anywhere in nature today of anything even becoming remotely close to developing itself chemically like RNA DNA does. Nothing. There's ideas that, there's certain organic molecules that landed on asteroids somehow surviving reentry and all that good stuff. But the reality behind that is there's only two of the four known chemicals that exist in biology were found on these asteroids, for example. None of the other theoretical processes to create the rest of the chemicals, much less the information embedded for these computer programs, exist. You would, I would have to think as an intelligent person, as a software developer, hardware developer, for this to have been able to do itself, I would have to have examples of it. I would have to have examples of something similar to a rocket engine developing itself out of rocks somehow.
Okay, now you're bringing up a really important point because I think a lot of people have been deceived by this idea of like we find organic chemicals on asteroids. And so that gives them, when we hear organic, it gives the impression of life.
Yeah.
So can you explain what is it that we're finding on the asteroids and why is that nowhere near close to any living system?
Yeah, the only difference between inorganic and organic chemistry is whether the chemical is used in biology or not. That's it. Otherwise, chemistry is chemistry, right? And it'd be something on the line of finding, somehow, I don't know if you could preserve a binary code bit on an asteroid or something, some sort of binary code, found naturally somewhere, and it combining with other, random binary code somewhere and making your computer by itself. It's worse than that. I mean, despite these nucleotides being in all the other pieces of the nucleotides themselves, because nucleotides are another complex chemical molecule combination, much as DNA itself is a very fast. Oh, that's another thing we could talk into, how DNA requires how zippers work. You got the different little strands, whatever. If you don't have a clamp at the bottom of it, the base of it, you can zip it up and they'll connect, but then they'll pull themselves apart unless you've got a clamp, right? DNA is exactly the same chemical wise, right? The whatever ribosome, nucleosome, whatever DNA, I forget what DNA is called. It has these oxygen and carbon backbones that are basically like zipper things, and you got the oxygen and the hydrogen and the what was the other one called? OH groups. It's been a little while since I've had to look at the basics of this. Where basically the zippers have other chemical forces to hold them together, but if you don't have a clamp, you have to have a starting clamp in the DNA chain. The DNA will just pull itself apart, and life won't exist. So where does that clamp come from? There's all kinds of wonderful theories out there, but it doesn't happen naturally. We can artificially build DNA molecules. Most life, even the most simple life, like we talk about, have millions and billions of bits of code. We can create maybe a couple of hundred in the lab, but we have to artificially, through nanotechnology, create a man-made clamp to hold it together, even temporarily. How DNA supposedly lasts, millions of years now, people are saying. It's like, what the heck? How? Show me. No one shows me, you know? So all kinds
Wow.
of things that go against I think it's little mythology. People want, they're biased. Like we talked about, they want a certain thing to be true. And so they'll come up with no matter what other fantastical claims. These code bits being found on asteroids. The guy that developed that theory, Sir Fred Hoyle, excuse me, he's one that's credited for the panspermia theory. He himself, in the 1980s, once he started to realize where all this stuff led, I've got his quote he published it in Nature, how this proves God exists. He said, "There must be a God," was the title of his article. He published this both in Daily Express on August 14th, 1981, and November 12th, 1980, in Nature on page 105, titled "There Must Be a God." He said, quote, "It is quite a shock." And this is the guy who developed the panspermia theory that we just talked about. "From my earliest training as a scientist, I was very strongly brainwashed to believe that science cannot be consistent with any kind of deliberate creation. That notion has had to be very painfully shed. I am quite uncomfortable in the situation and the state of mind I now find myself in, but there is no logical way out of it. I now find myself driven to this position by logic. There is no other way in which we can understand the precise ordering of the chemicals of life except to invoke the creations on a cosmic scale. We were hoping as scientists that there would be a way around our conclusion, but there isn't." Again, this is the early 80s. So,
Wow. And look how far we've come since then. And
yeah,
things are only worse on the
yeah.
material processes side now.
I was still in junior high, I think, back. And yeah, this work I've been blessed to have is like, there's just no way. It's like what Hoyle thought he knew is nothing compared to it. We are literally, Rebecca, not figuratively, we are literally uploading and downloading our own human design machine learning code in biology. Literally, there's
And
machine learning code in biology. We are downloading it to study and improve our own.
wow. And now you said this was published in Nature.
Mhm.
That's amazing. I can't believe that that statement was made in Nature. I just can't imagine
Yeah.
seeing that.
Well, he used to be a prominent scientist, in the atheist scientific community and stuff. He developed stellar nuclear synthesis. He developed what's called the carbon-12 formation theories. Um, so many things, not just panspermia. He was he was one of the in guys, the popular crowd, and it clicked with him one day. I was like, this doesn't happen by itself. There's no way, And so he was excommunicated, I guess. So.
Wow.